I'm pleased to introduce Abid Khan, who is currently at UNC Chapel Hill, right down the road from me, doing his postdoc in the Brian Straw Lab. Prior to that, he did his PhD a little bit west of here in UNC Charlotte. And it's been really great to be in the same area as Abid and get to know him a bit. And he's been doing some really cool work in the Straw Lab, including work that has led to him being the PI on a Lineberger Cancer Center Developmental Award, which gave him $50,000 to keep some projects going. And I'm pretty sure Abid will be hitting the job market any day now. So if you or anyone is looking to hire a fresh PI, reach out to Abid after this. Abid, take it away and let us know about SETDV1.
Thanks, Jackson.
SETD2, sorry, not SETDV1.
All right, thanks, Jackson. Thanks for the intro, and thanks to everyone for joining and for sticking around. And I'm really excited to share the story that I've been working on for quite a few years now. It has come together really nicely, we think, where we've discovered a completely unexpected function for a well-known histone methyltransferase, which is SETD2, in maintaining genome stability. So let's dive in. And as probably most of you probably know, that SETD2 is a histone methyltransferase that binds to RNA Pol II CTD during transcription elongation and deposits H3K36 trimethylation in the gene bodies. And this mark is then recognized by several reader proteins that facilitate some important downstream processes like gene body DNA methylation, RNA splicing, and DNA repair, which has been quite extensively studied. Everything we know about SETD2 for the last couple of decades really has been through its ability to methylate histones and what that histone mark functions in, and also probably a couple of non-histone targets of SETD2. Yet, surprisingly, we know very little about the SETD2 protein itself, mainly probably because it's such a beast to work with. It's about 2,600 amino acid, and it has a set domain and an SRI domain that binds to Pol II. But as you can see, more than half the protein is this poorly characterized and highly disordered extended N-terminus. And we heard some really nice talk from Henkel about disordered domains and how they function in chromatin. But this N-terminus is lacking in lower organisms like ESET2, which really begs the question, perhaps during evolution, the mammalian SETD2 acquired this N-terminus to perform additional functions than just histone methylation. So that's what I started to ask, was there more to SETD2? Are there more other functions besides just histone methylation? And to answer that, I had to first map out SETD2's intractome. And I did that by using the APEX2 system, where you do the biotinylation in the cells and followed by mass spec. And these are the results from the mass spec. And here's our bait and our positive hits of Pol II subunits. And when you get all of these ton of hits, when you break them down by GOTRM analyses, you see the usual suspects of chromatin-related proteins, transcription, mRNA processing, which was not really surprising. But what really caught my attention was this class of protein called nuclear envelope, which contained a lot of the nuclear lamina-associated proteins, like lamins, both A and B types, and LAB2 and emerin. But at first, it was really surprising and confusing how SETD2 would be interacting with these proteins because of what I knew about SETD2 versus the lamina. And lamina is at the nuclear periphery, where heterochromatin is tethered and lowly transcribed genes are present there, versus SETD2 is present on actively transcribing genes, which are thought to be towards the nuclear interior. So that was really confusing at the beginning. So I asked, when and where does this interaction occur inside cells? And so to address that, I did normal simple IF with SETD2 and lamina antibodies. And as you can see, sure enough, there wasn't much colocalization between SETD2 in green and lamins in red. during interphase. Then recall that our lab had previously shown that SETD2 levels actually peak during mitosis, which clearly indicates that there should be some important function of SETD2 during this phase, but we didn't know at the time. So I focused my attention on mitotic cells, and voila, I saw perfect, very good co-localization between SETD2 and lamins throughout mitosis, which indicates that probably the interaction is occurring during mitosis. And I confirmed this by performing proximity ligation assays between SETD2 and lamin. And for those of you who are not familiar with this assay, each green dot here is an instance of interaction. So as you can see in mitotic cells, we see a higher frequency of interaction compared to the interphase cells. So this clearly argues that SETD2 and lamins interact during mitosis. But what is the functional significance of this interaction? Since any perturbation in lamin biology, be it their expression of lamins or mutation in lamins, are known to cause nuclear morphology defects, I asked a simple question: what happens to nuclear morphology when we get rid of SETD2? And we've done this in several cell lines, and I'm showing you just a couple here. These are human cells, and these are mouse fibroblasts. As you can see, the control cells, you have nice and smooth and round nuclei with lamins at the periphery. But when you deplete SETD2, you get this spectrum of different phenotypes, like micronuclei right here. In fact, you get multiple micronuclei sometimes. You get invagination of the lamina, where lamins have sort of invaded towards the nuclear interior. And you get a lot of these blebs, and also just grossly misshaped nuclei, in addition to chromosome bridges and binucleated cells. So all of these phenotypes have been sort of previously studied in the context of laminopathies. So this clearly argues that there is some defective lamina. But what is the mechanism? How is SETD2 loss causing these phenotypes? And to answer that question, we have to first understand some lamina dynamics during the cell cycle. So as I'd mentioned, nuclear lamina is this highly filamentous meshwork of lamin proteins that is present at the periphery of the nucleus during an interphase cell. But as cells progress through mitosis, the nuclear envelope has to break down. So does the nuclear lamina. So the lamina has to depolymerize. So the lamins have to disassemble. And that depolymerization is carried out by the action of a major cell cycle kinase called CDK1. A CDK1 hyperphosphorylates lamins, all lamins, A types and B type. And this causes them to disassemble. And by the end of mitosis and going into G1, lamins get dephosphorylated and reassembled into the lamina. Now, this cycle of phosphorylation and dephosphorylation is really critical to maintain the genome integrity and to maintain nuclear morphology. So since SETD2 and lamins interacted during mitosis, I wanted to understand get some more temporal sort of insights into lamina dynamics during cell cycle. So I performed live cell imaging with GFP tagged lamin A. And as you can see in control cells, the lamina is nice at its nuclei round and lamina is intact. But as cells enter mitosis, the lamina starts to depolymerize, and by metaphase, lamins have completely depolymerized. But what's interesting is when you lose SETD2, you see, I started noticing that these partially depolymerized lamin filaments that remained throughout mitosis, which indicated that the phosphorylation was probably not going to completion, and therefore lamins were not getting depolymerized completely in the absence of SETD2. And if that is the case, and if SETD2 has a direct role in this, then we should be able to conditionally or acutely deplete SETD2 right before mitosis and observe what happens to lamin phosphorylation. And to do that, I generated SETD2 DTAC cell line and synchronized them and monitored the levels of laminin phosphorylation at two key residues, serine 22 and serine 392, as cells progress through mitosis. So in control cells, you can see these cells reach mitosis around at the 12-hour mark, and you see a dramatic increase in laminin phosphorylation levels at both these serines. But when we lose SETD2 immediately prior to mitosis, we see a significant reduction, about 50% by mass spec. We see a significant reduction in both these serines, phosphorylation at both these serines, which clearly argues for a direct role for SETD2 in regulating laminin phosphorylation during mitosis. Now, the phenotypes that we see upon SETD2 loss, are they due to a defective laminin phosphorylation during mitosis? So if that's the case, then if we were to mutate these serines into alanine such that they can no longer get phosphorylated, we should be able to replicate or phenocopy SETD2 loss. And that's what we see when we mutate both these serines into alanines. We see a similar range of phenotypes that we see when we lose SETD2, arguing that SETD2 loss causes these defects because of reduction in laminin phosphorylation. Okay, so now the big question then was, what aspect of SETD2 is regulating this phenomena? And since SETD2 is an enzyme, so the low-hanging question was, is it the protein or its catalytic activity that's important? And to test those two questions, we made use of this system where we have the SETD2 gene, which is heterozygously floxed, which allows us to conditionally knock out just one copy of SETD2, thereby depleting just 50% of the protein, which is important in the context of kidney cancer because 90%, greater than 90% of kidney cancers or CCRCCs lose one copy of SETD2, leading to SETD2 haploinsufficiency. So then we asked what happens to nuclear morphology when we lose one copy of SETD2? And surprisingly, if just losing one copy of SETD2 can cause widespread nuclear morphology defects that are similar to a complete loss of SETD2. Now, the next main question was, is its catalytic activity that is important? And to test that, we used a very highly selective SETD2 inhibitor, which wipes out K36 methylation here, but doesn't impact laminin phosphorylation. And much to our surprise, it did not also affect nuclear morphology. Now, these data tell us two things. One is that SETD2's protein levels or dosage is highly critical for this function. And second is that its catalytic activity is completely dispensable. Now, as far as I know, this is the first report of a non-catalytic function for SETD2. Okay, so if it's not the catalytic activity, then what about SETD2? Which part of SETD2 is contributing to this function? To answer that, I generated a series of mutants where I mutated the SRI domain or the SET domain, and I also chopped off the N-terminus. Again, going back to my original question, does the N-terminus play any function in SETD2 biology? Now, in the interest of time and for simplicity, I'm just going to summarize a lot of the data here. What I first looked at was K36 methylation. And not surprisingly, the SET mutant didn't rescue it, but T-SETD2 was able to rescue. It has been known in the field, this C-terminus half of SETD2 can rescue K36 methylation. But what's more interesting was T-SETD2, or the truncated SETD2, failed to rescue laminin phosphorylation and also failed to rescue all the nuclear morphology defects, suggesting that the N-terminus is where all the action is, and N-terminus is what is contributing to this nuclear lamina regulation function of SETD2. But what about the N-terminus? How is it doing it? So we took an unbiased approach. We tried to map out the interactome of both the full-length enzyme versus the truncated version. And here on proteins enriched on the wild type. So what we noticed was that a lot of all the lamina-associated proteins were enriched on the full-length enzyme. And not just that, the kinase that was responsible for lamin phosphorylation was also enriched on the full-length enzyme, which sort of suggests that the N-terminus is perhaps acting as a landing pad for a lot of these multi-protein interactions to occur, and probably as acting as a scaffold. Now, if that model is true, then if we remove the N-terminus, the interaction between CDK1 and lamin should reduce. So we tested that by performing PLA assays, proximity ligation assays between lamin and CDK1 in cells that are expressing either full-length wild-type enzyme or the TZD2, the truncated ZD2. And indeed, we see a dramatic or significant decrease in the interaction frequency between lamins and CDK1. Okay, so the next question was, since the N-terminus is about 1,300 amino acids, we wanted to understand a little more in detail as to which part of the N-terminus is doing this function and how is it doing it. So I broke the N-terminus down into 3 different sections, 3 different fragments based on their amino acid composition. And I deleted each of these regions and performed IF to see which of these regions can rescue nuclear defects. Interestingly, deletion, the region A was completely dispensable. If you delete A, it can still rescue nuclear defects. Deletion of B and C individually caused minor defects, but it's the deletion of both B and C that phenocopied either a complete loss of ZD2 or have loss of N-terminus, suggesting that B and C together acting cooperatively to regulate this function. Now, how are these regions doing it? Can they interact directly with lamins and CDK1? And can they influence lamin phosphorylation in vitro? Those were the main questions we had. And so to address that, we had to purify these fragments, recombinant proteins, and which was really a pain. But in the end, we resorted to flanking these disordered proteins with two highly soluble domains, like SUMO and the B1 domain of protein G, which made it easier to express and purify these fragments. And then we asked, can these proteins interact directly with CDK1 and lamins in vitro? And long story short, the answer is yes. Both B and C can directly interact with CDK1, with the C fragment interacting more robustly than the B. And we see the similar things with the lamins as well. Now, the million-dollar question, and my favorite part of the paper, was can ZD2 influence or enhance lamin phosphorylation in an in vitro kinase assay? So pure biochemistry, all that I've shown you before is if we lose ZD2, we see a decrease in lamin phosphorylation. Now I'm asking the reverse question, if I add ZD2, can it increase lamin phosphorylation? So to do that, I did a simple experiment where I mixed lamins and CDK1 together, split that into 2 reactions. In one tube, I added a control protein, which is SUMO GB1, and to the other tube, I added ZD2. And I added ATP and took time points and assayed phosphorylation levels of lamins at 2 different residues. I'm showing you here the control reaction, and at 40 minutes, you see this much level of phosphorylation at CD22 and 392. However, if you have ZD2, the C fragment, phosphorylation of these 2 residues increases dramatically, suggesting that ZD2 is directly playing a role in enhancing lamin phosphorylation by CDK1. Now, I think I'm running short of time, so I'll just run through these slides. We asked what happens, is this required for human disease like CCRCC? And indeed, that's what we see. If we put back a copy of ZD2, we can restore nuclear morphology and also rescue tumor growth in xenograft model, which is, in fact, if we just add back the BC containing region, we can rescue tumor growth. to summarize, I've shown you that SETD2, we've identified a new function of SETD2 that is in mitosis, independent of its role in of catalytic activity. And with that, I'd like to thank my advisor or mentor, Brian, and all the undergrads that have worked with me over the years. Thank you.
Thanks so much, Abid. It's such a cool story. We have one question in the Q&A. I'll go ahead and ask one thing first as well. I'm wondering how you think these interactions between this region of SETD2 is occurring with CDK1. Like, have you tried like plugging it into AlphaFold to see if they like adopt a structure to interact, or do you think this is like a condensate or an IDR driven interaction?
We have no idea, and this is even in CDK biology. It's not known. There's no precedence for another protein to interact with CDK1. There is CKS1 that increases its multi-site phosphorylation intrinsic activity, but a disordered protein interacting with CDK1, it's not known. So that's what part of my proposal is, to look into this interaction. And we tried doing AlphaFold. We couldn't get anything because of SETD2 is completely disordered. But I want to break it down into smaller chunks of SETD2 and see how that interacts, and probably then do AlphaFold and see how that interacts.
Cool. And then quickly, we'll do this question again from Maria. She says, you've nicely shown that the N-terminus is required for the interactions with lamins and their phosphorylation. Is it sufficient for those activities, or do you think you need coupling of the N and C terminus for normal lamin biology?
That's interesting. But what we've seen is with just the T-SETD2, which lacks the N-terminus, it can still methylate all histones exactly like the full-length protein. So we know it's still going to the regions that it's supposed to because of this interaction with Pol2, and it's able to methylate gene body, do histone methylation. Whether we need coupling of N-terminus and set domain for other functions, maybe a non-histone target, others, we don't know that. And that's an interesting point.
Awesome. Well, since we are hitting the hour point here, I'd like to again thank all three of our speakers for this great session. If anyone else has further questions, I encourage you to reach out to the speakers individually or join the Fragile Nucleosome Discord, where you can start a conversation there yourself.